Greetings to everyone. Today's topic is about microbial production of vitamins and enzymes, presented by Vijayalakshmi R, 2nd MSc Microbiology, Sacred College, Tirupattur. Microbial production of vitamins. Vitamins are organic compounds that perform their specific biological functions or for normal maintenance and optimal growth of an organism. Vitamins cannot be synthesized by higher organisms, including man, and therefore have to be supplied in a small amounts of in the diet. Microorganisms are capable of synthesizing the vitamins. In fact, the bacteria in gut of human can produce some of the vitamins which is appropriately observed can partially meet the body's requirements. It is an accepted fact after administration of strong antibiotics to humans which kill bacteria in gut. Additional consumptions of vitamin is recommended. Microorganisms can be successfully used to chemical productions of many of these vitamins, example thiamine, riboflavin, pyridoxine, folic acid, and pantothenic acid, and biotin, vitamin B12, ascorbic acid, P-carotene, provitamin A, ergosterol, provitamin D. Fat soluble vitamins. Vitamins are provitamins A. In our bodies, these provitamins can be converted into retinol and retinic acid, which are active form of vitamin A. Vitamin A is group of a different retinols, retinic acid, and retinol esters. Provitamin A is composed of various carotenoids and most important one being beta carotene, and while others are alpha carotene and beta tryptophan. Vitamin A is involved in many functions such as immune system, vision, reproduction, cellular communication, cell growth, and differentiation. It is crucially form of the normal development and maintenance of heart, lungs, kidneys, and other organs. It is essential for vision since it is key component of retinal receptors. In the diet, both vitamin A and provitamin A can be obtained from food. To perform vitamins form are currently available in daily consumed. This is a structure of vitamin A. Some microbial processes have been put of essential use using the green microalgae of Dunaliella and the fungus of Blachyelsa trispora to produce beta carotene. And certain conditions of Dunaliella can accumulate more than 0.1 gram of one cells, dry cells 20 to 30 percentage of salt, nitrogen limit 10,000 lux and 25 to 27 degrees Celsius for 3 months. And wheat trispora fermentation can produce 2 grams of dry cells after 7 days. Whole cells used to directly feed addictive. Alternatively, beta carotene can be purified from then. Candida utilities and Saccharomyces cerevisiae have been modified to express bacterial keratins and biosynthesis genes affording beta carotene titers, vitamin 01 and 04 mg dry cells. E. coli has also been bioengineered to produce the carotenes using different strategies in which the expression of genes from Enterococcus faecalis and Streptococcus pneumoniae is the most productive, reaching titers of 460 mg and 1.1012. Vitamin D. Vitamin D is a fat soluble compound from cholesterol and ergosterol. On the one hand, cholesterol is modified into metabolically to produce 7-dehydrocholesterol and which can be cleaved by UV radiation from kilocalciferol. Neither the D2 nor the D3 form is active and humans they must undergo two hydroxylations. First, liver converts vitamins D into 25 hydroxy vitamin D calcium, and second the kidney transforms into 1,25 dihydroxy vitamin D. Normally vitamin D and D3 are used in food and feeds. This vitamin is essentially it's also calcium absorption and permits the normal mineralization of bone. It also modulates the cell growth and neuromuscular human inflammation functions. Vitamin D can be synthesized the most people through exposure to sunlight. Excessive radiation can be carcinogenic, so it is prudent to the exposure of a skin light complete diet, including this vitamin. This vitamin can be found naturally, mostly in fleshy or fatty fish and liver oils. Most small amounts are also present in beef liver, cheese, egg yolks, and some mushrooms. Currently, it is common is to satisfy the need for this vitamin by supplementing its level in foods to create fortified foods vitamin D. Ergostrol, its producing bioprocess including generally recognized as safe yeast of Saccharomyces cerevisiae and Saccharomyces eubaram and C utilizes 10 to 30 mg gram of one dry cells. Fungi such as Trichoderma, Cephalosporium, and Fusarium have also been investigated with regard to their capacity accumulate ergostrol, afro lower production titers, and Saccharomyces cerevisiae. Saccharomyces cerevisiae have been bioengineered to increase the accumulation of ergostrol by overexpressing different enzymes involved by synthetic pathway and using molasses and cheap carbon source. In addition, strains producing cholesterol instead of ergostrol have been developed by metabolic engineering of Saccharomyces cerevisiae. Vitamin A. Vitamin E is the vitamin E group of formed by different molecules with antioxidant activities. There are 8 chemical forms, 4 tocopherol and 4 of tocotrienol. Tocopherol is a fat soluble antioxidant involved in the regulation of reactive oxygen species. It's producing during a fat oxidation, thus it's actively diminishes the damage caused by the free radicals to cells, such as free radicals being able to lead to the develop of cardiovascular disease or cancer. Natural source from higher plants. Higher plants, peas, pinifera seeds, soybean oil, rice bran, wheat gram grain, sunflower, and then vitamin E can be extracted by exon or supercritical fluids. The main drawbacks of this source are their low contents tocopherol and generally low proportions of alpha tocopherol. As an alternative to this approach, photosynthetic microorganisms are known as accumulated tocopherols and Eugellina calyzals have been seen to be the best producer organisms reaching 7.35 mg and dry cells where 97% of tocopherols and alpha is formed. Its major limitation is the culture conditions. It is also easily contaminated proposed alternatives. It's Dunalilia and Spirulina. Water soluble vitamins. Vitamin B1, also called thiamine, it's water soluble compound and their five phosphate derivatives including thiamine monophosphate and thiophosphate, diamine triphosphate, thymine triphosphate, and adenosine thymine triphosphate. Thiamine has essential metabolic function. Its deficiencies of this compound are associated with imbalances in carbohydrate status because it is involved in oxidative decarboxylations and transkeltolysis reactions. Cornybacteriaceae is a bacteriaceae in which of microorganisms containing a mutation that deregulates thiamine production and cause thiamine products can be released from the cell. Vitamin B12 is produced chemically to supply human and animal needs over time. Two methods of synthesis have been developed. Consideration of pyridamine and thiosyl rings. Constructions of thiosyl ring 1 are performed by the remain portion. Several strategies overexpression and deregulation of the genes involved in precursor synthesis and pathway engineering, among others, have also been described. Vitamin B3. Vitamin B3 is a group formed nicotinic acid and nicotinamide and other compounds such as inositol hexanicotinate that exhibit relative biological activity. These compounds can be clustered to under niacin like all B vitamins, niacin, and involved energy metabolisms. For healthy skin, hair, eyes, liver and nervous system. It is used to or currently under investigation for use in therapies high cholesterol, atherosclerosis and heart disease, diabetes and osteoarthritis. The vitamin can be found in different foods but can be also produced from the essential amino acid tryptophan, which can be obtained from most source of protein. Chemically, it is synthesized by oxidant and 5-ethyl methylobutadiene, and by total hydrolysis nitriles have been overexpressed Rhodococcus rhodochrose, generating a strain able to convert almost all three cyanophyridine into nitronic acid. Nitrile hydratase in our Rhodococcus exceeded 50% of the total cellular protein, permitting high production of neoectin. Both reactions are mole host. Even very high concentration of the substrate are used to no significant. Vitamin B5 is also called pantothenic acid. Commercially, it is available as D-pantothenic acid as well as exopantothenic and calcium pantothenic, which are chemicals made in the laboratory from D-pantothenic acid. Vitamin B5 has a role in the breakdown of carbohydrates and fats energy. This vitamin is also involved in the production of red blood cells and stress and sex-related hormones and maintaining a health digestive tract, helping to obtain other vitamins. It is also essential for synthesis of cholesterol and regulates fats in this blood. The fungal enzyme lactohydrolase is the key protein for racemic resolution and dipantonic substrate of not I pantolic tone. The hydrolyze dipantolic into dipantonic acid, which is easily separated from the teloenteromere of pantolectin. High activities this enzyme have been reported in Gibberella and Cylindrocorphone and Fusarium, and cells of Fusarium oxysporium have been immobilized calcium alginate cells. Candida pharoxylosis cells have been proposed for biotransformation owing to the high activity of carbonyl reductase. A variation of this method is the hydrolysis of ketanophantolactone to be ketofanoic acid, which is reduced by agrobacterium species introduced by pantonic acids. Microbial production of enzymes. All enzymes have been infrastructural backbones of protein. In some enzymes, only protein exists, while in others, covalently attached carbohydrate groups may be present. Often, these carbohydrate groups may be play no part on the catalytic activity of the enzyme. Through they may contribute stability and solubility of the enzyme. Metal ions known as cofactors and low molecular weight organic compounds known as coenzymes may also be present. Cofactors and coenzymes are important for the stability and activity of the enzyme. Semi-solid medium. The system known as koji or moldy bran method of solid-state fermentation is widely used in Japan. This medium consists the most sterile wheat rice or rice bran acidified with HCl. Mineral salts including trace minerals are added. An inducer is also usually added. 10% of starch is used for amylase and gelatin and pectin for protein and pectinase production respectively. The organisms used are fungi which appear amenable to high enzyme productions because of the low moisture conditions and high degree of aeration of the semi-soluble medium. The most bran inoculated with spores of the appropriate fungi is distributed with either a trays placed or revolving drum. The temperature of the bran is kept at about 30 degrees Celsius by the circulating cool air. The production period is usually 30 to 40 hours but could be as long as 7 days. Growth in semi-solid medium seems sometimes encourage and enzymes range different from the produced submerged growth. This Aspergillus oreaceae on semi-solid medium will produce a large number of enzymes, primarily amylase, glucoamylase, and protease. This moist bran inoculated with spores of the appropriate fungi is distributed either fat trays or placed in revolving drum. In submerged culture, amylase production rises at the expense of the other enzymes. If Aspergillus oreaceae producing a chemical powder and containing amylase and some protease is grown in submerged culture for protease components or protease components are formed, but this are less heat resistant than those produced in submerged fermentation. Most enzyme production fact by submerged cultivation in a deep fermenter. A submerged production has replaced the semi-solid production wherever possible because the latter is labor intensive, therefore expensive, where labor scarce, and because of this risk infections and generally greater ease of controlling temperature, pH, and other environmental factors in a fermenter. The medium must containing all the requirements for growth, including adequate source of carbon, nitrogen, various metals, trace elements, growth substance. The inducer may not always be the substrate of the sometimes of submerged productions. Enzyme extraction. The producers for the extraction of fermentation products described or applicable to enzyme extraction. Care is taken to avoid contamination. Stabilizers such as calcium salts, protein, sugars, and starch hydrolytes may be added. A destabilizing metals may be removed with EDTA. Antimicrobials, if used at all, are those that are normally allowed in food such as benzoates and sorbate. Most industrial enzymes are extracellular in nature. In the case of cell-bound enzymes, the cells are dispirited with before centrifugation and or vacuum filtration. The extent of the purification after the clarification depends on the purpose of which enzyme is it to be used.